Hi, everyone.
Okay, we'll just wait for a few more minutes. I think folks are still trickling in. While we're waiting, I know there was one team that had to finish their Fred Park presentation. I just want to cue who that was. Do you know, Abina? Oh, Divyanshi. Do you want to do your Fred Park today?
Just let me ask Alankrit also if he's joining, and then we'll do it.
Divyanshi, can you hear us? We don't hear you.
Hello, am I audible?
Okay, yes, I can hear you now.
I was saying ki let me just ask Alankrit, one of my team members, if he's joining, and then we can do it together.
Okay, yeah, so just text me if that's the case. Otherwise, I'll start on the threads that I was going to cover today.
Okay, just
yeah.
In a minute, I'm asking him.
Yeah, that's good. I always have to go to the source. And I don't see that source button usually here. Oh. Should I try another connector? Okay, nothing. Oh, I think we got it. So yeah. Now, which one is it? 2? Yeah. This current connector. Yeah, no, it worked. It worked. Okay. Yeah. Or it didn't work there, but I know it did blink here. Okay. So right before we start, questions, threads, I think on dates, that becomes very important. Let's just schedule the presentation dates. So what was the 2 dates? I think we'll probably need 2 days to cover. And I want to do that before the finals week. 14th and the 16th. So let's just mark that out for a second. I think the talks usually would be somewhere around 10 minutes. And then depending on number of teams signed up, we will do the length of the Q&A based on that. So yeah, let's just make sure everybody knows those as the dates for the final class. And then we should just post that on Discord as well, just so that it's valuable to remember working backward when you guys are all presenting. Okay, other logistics. Smriti, go ahead. Was there a question? No, okay, yeah. I'm assuming everybody has a team now. You guys are moving ahead. If somebody doesn't have a team, please urgently reach out to me or the TAs. That would be very difficult to contribute if you haven't joined a team as yet. So, and I think one of the threads would be is if we haven't had a one-on-one meeting, that's also urgent at this point. There's not going to be enough time. And at this perspective, one important thing would be is try figuring out the needs. So when we're sitting down one-on-one, there are some very clear, this would be a valuable thread to achieve by end of this quarter. And then we'll just work backwards from there to see what kind of resources people need. You know, for some of you, it might be just access to a prototyping space. For some of you, it might be help on CAD. Other folks, it might be an instrument that we've built in the past. So it depends on teams. Everybody is different, but it would be very valuable to make sure that you ask for what you might need as soon as possible. And for some of you, it's just more brainstorming or access to folks that work in different fields to be able to talk to people. There are lots of mentors in many different fields. The only challenge is that unless you ask, neither would we know, nor we would sense, a sense of commitment. So I don't want to point and assign a mentor unless somebody really very clearly makes that clear in how they're thinking, what the gap might actually be. Okay, so I think we're going to transition to covering one more platform and a case study of what we've been building in the lab, which is relevant for some of the assays that folks and threads here have been running. It's also related to optics, but it's much broader in terms of the instrumentation and instrumentation design. And I think the lab this week will also just focus on that as a platform. So I'm going to dive into that for a little bit. And then Divyanshi and Alankrit and any folks, maybe in the very end of this class particularly, folks can finish a Fred Park if they haven't done so as yet. So let me share my screen. So we had talked about most of you had okay, so I think I wanted to start here. Most of you in the past had seen the discussion on octopi and malaria, right? I think, although we haven't actually covered the mechanics of octopi either. Oh, I'm just realizing that. That's okay. I think, yeah, it's relevant. So one of the challenges that we started a couple of years ago, when I say couple, I mean 10 years ago, was really this idea that the way scientific instruments are designed per se are designed as black boxes, right? So in both the context of diagnostics and the context of just scientific instruments that you use in a lab setting, you usually would have access to one given thing that the instrument is able to do. And most of the time, the design is not open even to the scientific community itself. So that sort of started for us this journey in modular microscopy and modular instruments in general. And so this was the very first version of octopi. And maybe I'll kind of just walk through this picture for a second. But there's something very interesting going on in this picture in terms of just instrument design. Now that has become what we call octopi and squid as the two platforms. And the idea was to break down the key principles of this microscopy. There is detection. You need some amount of control and automation. You need some amount of compute. And you need illumination and bringing both light or other things to the samples. So this was, this is not the very first. And then, of course, on the right that I'm realizing, which is totally not visible there, was compute. That there is also the compute layer on the right. The compute was changing quite dramatically at that time itself as well. I don't want to take that. And so that leads to this framework, which we ended up calling, I think this became really the octopi and squid. And then the parts that you will see now have changed dramatically, but maybe on Friday, I'll bring some of these older versions itself. In the very first years of this work, maybe I would say 2014, 2015, much of the parts were also 3D printed, but we moved away from it just from a precision. They were at that time all magnetically coupled. So you can see 3 different heads for the same instrument that would just snap right here. So you could change the detection right away. Similarly, these 3 would snap at the bottom and you would change the illumination. And then of course, this you can clearly see as a module was the scanning. So this was sort of the dream. And we really stayed quite close to that dream. This is what that instrument actually looked like. And one of the things that will become obvious very soon is a big gap essentially exists between how you do automation in microscopy-based assays. Most of the time, what ends up happening is the cost of those platforms or just how reconfigurable they are is very limited because they were designed for that one specific thing. And I think even in terms of when we release this paper, for example, what this instrument is able to do compared to what you can do, say, for example, commercially, we were able to build something like this for around $1,000 in parts. You can see there are 1 or 2 3D printed parts in there, but everything else is just machined. And the heads are changing. So you can see right on the left, for example, is a very low mag objective from a reverse lens. But then that same approach actually allows us to just also put traditional lenses. And then I'm just going to show you some data. And I think maybe on Friday, you'll also meet Hong Chuan, one of the key graduate students. But this is what the instrument looks like. So this is fun to see. Now, this is, I would say, 2017. And now you'll see 2017, 2019. And I'm just going to run this video for a second. And what you're watching is essentially, this is 3x the actual spanning speed. But an object that big is essentially scanning a malaria slide. So we talked quite a lot about in the past on the challenge on malaria microscopy, the fact that you really need certain sets of people. This was our first answer. And then you can see the instrument primarily runs on a power bank. This was another one of the key criteria that we should actually be able to run this on a power bank. But the key really arises here of what you're able to do. And I think this is kind of, in general, why I'm spending enough time on this. And this week, we will try maybe assembling one of the squids, is because what you can do with these types of approaches. So now we're going to see the data that that instrument just collected. So what you're looking at is essentially an entire slide. So the scale bar on that is 2 millimeter. And I'm just going to play this to give you the sense of the amount of data that's collected on a single patient. So those are individual parasites now. You can see platelets right there. And then I'm just going to zoom out, and it gives you the framework of, in this single image, there is roughly around 20 million cells. And traditionally, when you were to sit down, for example, if you were to sit and do this as a human, and you spend half an hour or so, you would essentially be looking at a square that might be that big. That's roughly around, I would say, 3,000 to 5,000 cells. And so this is the big gap. And essentially, for very straightforward instrumentation, you're able to just access the kind of data that you wouldn't even think about. This is also exactly the amount of data that you can from a single single drop of blood. So this is a finger prick blood that has roughly around 20 million cells. And I think one of the things that becomes, when you guys looked at Inkwell, for example, this was the smear that makes something like that possible. And the even more exciting is really essentially what you can do with this data. So this is just one example of what we do with a data set like this. and I'm just going to play this for a second. What we have done at this point is taken those data sets and segmented them. And this is actually from a platform that uses a $5 lens. So at that resolution, so this is fluorescence channel, this is bright field or phase contrast, and this is essentially a more expensive, I would say, $1,000 lens. And the key point is that, of course, in bright field, you don't see anything, right? There's not enough resolution. That makes sense. But in fluorescence, you actually see the correlation. And one of the key things that you're able to do when you're building and designing sets of instruments is not just depend on morphology. So traditionally, the way malaria detection is done is you primarily look for morphology. And even with this, I think you can start seeing, so I'm just playing essentially all the parasites in that have been identified and they're aligned to the center there. But the key bit here is that you're seeing a color shift. So there is green and this is blue. And so what that current instrument, Octopi, is doing is it's actually doing spectroscopy rather than just microscopy. So what I mean by that is when you illuminate with the given light source, we're essentially looking for a very broad range of fluorescent light coming out. And for a reason that we discovered throughout this process, we are able to build and design something where the parasite is going to show up. And actually, this is very clear for why. Maybe before I mention, anybody wants to think about, look at this image on the right and one on the left. This is a platelet. This is a parasite. And actually, it's very interesting. There is a little tinge of green even in a red blood cell. So anybody wants to sort of guess and think about why would there be that the parasite lights up differently? This is a nucleic acid stain. So this is DAPI, and we are essentially staining the nucleic acids inside a cell. So first of all, why is there nothing in the red blood cell? It doesn't have a nucleus. It's a very special cell. Red blood cells don't have a nucleus, and a lot of diagnostics depends on that. Rare chances there is a nucleus in a red blood cell. That's when you have a mistake when the red blood cells are being made if a nucleus gets put in. So that's good. That's why even in such a massive background of cells, you're able to detect something. So now let's move to platelets. Platelets are lighting up as blue. And so the answer to, I mean, again, this would also be true for white blood cells and others. They will all have a nucleus. They will light up. And that makes sense because platelets do have a nucleus. So now the question is, why is the, and this was it took us a long while to figure this out. This is actually was very counterintuitive and very exciting. It's a serendipitous discovery that had not been made before. Why would it be that the parasite would show up as green? And what was very interesting is in the past, people had not seen it. And only because we were very frugal, when Hongshuan was putting these things together, I didn't want us to buy $1,000 or $1,500 fluorescent set because the whole platform was supposed to be less than $1,000. So fluorescence filters are these filters that you buy that allow you to cut a given, essentially, if you're going to illuminate something with a given light source and you're looking for another wavelength that you believe it's going to emit, you essentially, a filter set cuts off all other light other than that specific wavelength, because you want to be able to not see the light that you already are shining on it. And so these are called bandpass filters, and those bandpass filters are really, really good. So they cut off exactly around the wavelength that you put in. But that requires you to assume that this fluorophore is going to be shining at this specific wavelength. And so now I think there's a clue in that picture. Anybody wants to guess why does the parasite first of all, it doesn't just look green, it does look blue as well, right? But overall picture from that is there is a lot of green in that signal. So what else is there in a cell that could get stained with, which is also nucleic acid? RNA, exactly. So we were accidentally staining RNA. And then it turns out that the way this dye, DAPI, binds to RNA, it actually emits a completely different wavelength of light. So now what we are measuring by using a filter that was not so good, we allowed all the light to come in and we actually uncovered something that was not so obvious. We're measuring the ratio of DNA to RNA in a given cell, which can then be a signature. And you can see in these platelets, there is very little RNA in these sets of cells. And actually what's very interesting, in very young blood cells, you also see some amount of RNA. And that's this background green signal that's also showing up in the RBC itself. So this one insight completely changed the game for us for what we could do for malaria, essentially, because, yeah.
Where is the RNA coming from?
Oh, just the RNA is essentially everything that the nucleus needs to tell the cell to do. It can only, its language of talking to the rest of the cell is just through the RNA. So every protein, everything that has to be made in the cell, it first has to be transcribed in a form of an RNA. And so what you're actually watching is that's the cytoplasm of the cell, and then that's the nucleus in one corner of the parasite. And then I'm going to see if I can find one or two more pictures of this. Just yeah, I think they do. And you could see a small amount of it in the background, and especially when they are younger. So I'm going to see if I was just assuming that I've shown you all this data before, but maybe not. So now this is the current platform. And one of the threads that you can now see that the data and the background is much cleaner. Again, you can see parasites. You can still see that spectral shift. Of course, this is not absolute because depending on how I'm choosing to display it, but the big thing to see is that that's the platelet and that's the parasite. And effectively, this is the current system. This resolution is what we are starting to deploy. And this is what the deployment of this. So first of all, the current system looks like this. It's a little bit more in terms of the instrumentation side. We have closed it up quite a lot because there's a lot of dust and other things where we work. It has now, the fluorescence is all, you can see this is the LED. You will watch this when we assemble one of these together. So this is the illumination path. That's the detection path. We now use these cameras that are used essentially in security footage. Those are some of the best cameras that are out there at this point. And this is the actual physical instrument for what it looks like. It's grown to be a little bit bigger, maybe that big. And the reason I'm just sharing this is I'll talk a little bit about once we enabled these sets of platforms, we started building lots of other applications. Of course, malaria was the first one. Now the same tool can actually do TB. Currently, we are working on another version for this for schistosomiasis. And the goal is that once it's installed in a primary health center, there could be like apps on your phone. There's a common hardware, but many different applications that could be built on top. And that's all in the firmware. And so I think for folks that are interested, at this point, there's roughly around 25 of these instruments that we have. And maybe I'll see if I can find, yeah, maybe this is one example of the field site. So this is the primary healthcare center in Kédougou in Senegal, which is one of our big sites. And this would be essentially a clinical location. This is so effectively, this is where one of the machines is installed, and you can see this is the traditional microscope. Many of you have seen that video that I shared in the past. This is again what the, I'll see whether this actually, yeah. So just to give you a context of, most of these places are fairly difficult to reach, and the instruments have to operate in a scenario where we can charge them overnight, but then they have to operate on power bank that is essentially for a whole day. And we start to realize the inaccessibility is a huge part of the technical challenges. So, you know, the robustness of these instruments matter quite a lot. And it just gives you a context of the inhospitability for instruments for a place like that. Okay, I think any questions on Octopi before, because I wanted to spend a lot more time on squid today, not realizing that I hadn't finished the threads on Octopi. But I'm curious, I'll search if I can find one more image that I want to show you guys. Any questions? Yeah, go ahead.
Question. Is there any scanning happening in this, or?
Yes, I'll play that video one more time while I look for. So watch what's going on. The slide is in there, and you should be able to see the stages moving in X and Y. So the entire thing is only possible because we can scan very rapidly. So every field of view is collected, and then that field of view is, I'll see whether there is another video. So you can see the slide that goes in, and then that slide is scanned over the entire space of the smear itself.
I see. Are you limited by the scanning speed of this setup, or is that not a limiting factor?
No, it used to be in the past. Our goal essentially around for malaria is go from patient to diagnosis in 10 minutes. 5 minutes we dedicate for scan time, and the moment scanning begins, we essentially do spot detection, and the compute starts in parallel. And the idea is to be able to tell within a period of 10 minutes the parasitemia. Usually, if the parasitemia is very high, within the first few images, you can already tell. But the goal is for a negative patients or very low parasitemia to be able to tell whether you are positive or negative. And so I think one way of thinking about this is, this is kind of, first of all, this is that difference that you can see now plotted in a scatter plot. So you can start seeing the huge variation associated with those. But one of the things to think about in terms of classification accuracy just comes from the fact that for a high parasitemia, you don't need that many number of samples before you tell whether somebody is positive or negative. But in low parasitemia, you need to scan more number of slides. So I think one thing that we are thinking about in our approach currently, actually, this is a fun comparison between 2019 and 2021 in terms of the optical configuration. So some important things to also just notice in terms of changes, which become important when you deploy something in the field. In the setup in the 2019 paper, we were using essentially just these very cheap UV lasers, and it significantly reduces the cost. As you can see, there is no optical arm, there is no cube, we save on that. But the practical reality was, so I don't know if you guys can see my cursor right here, right there. The reason we moved away from this was the mounts, any little bump or a bang that would happen on the instrument misaligns the laser. And it was painful enough after deploying them many number of times that a little bit of that cost was actually worth to make the instrument robust for the kinds of conditions that we care about. And so eventually we did move to a sort of a cube-like configuration for robustness. And again, this is a trade-off that can only be learned on paper. This saved and shaved a lot of costs. And again, both of these versions are on bioRxiv. People can go take a look at the current design. And I think I'll walk through another big thing that we have changed between 2021, 19, and or this started in 2017 is you can see these motors. I'll bring these motors on Friday. So these are linear scanning motors. We borrowed them from DVD players. DVD players are basically the best scanning instruments that are deployed because of video game industry that's out there. So it's actually quite valuable. But then we also realized that there was a certain amount of stick slip and we can't afford a miss over time, and just they degraded over time in terms of just the amount of scanning that happens in here. And so you'll see now we have switched to the NEMA motors, and then we'll talk a little bit about this also on Friday. So I think, as you can see, that some of the setup has changed, but driven much more by the current needs. And then effectively, maybe the last thing I want to mention is, so this is the spectral shift, now pseudo-colored. And you can essentially see that spectral shift right there. So that yellow peak, this is the RNA peak that's coming. And that's what I mean. The deep blue here is the excitation peak. This is what we're exciting at. Then suddenly you see the next one that's coming from the DNA. That's the green curve. And then the yellow curve here is really the significantly shifted RNA peak. And you can actually see some of that even in the parasite. You can see the DNA and you can see the cytoplasm, and then you see the cytoplasm light up. So this the shift is quite subtle. It's on tens of nanometers, 20 nanometers, but that's sufficient primarily because the detectors are also fairly good now. And again, this is the sensors are not scientific sensors. They're just commercial security cameras, and they're sufficient. Okay, so I think that's all I wanted to say on, I guess maybe I'll just mention two more threads on this front, is the deployment side of the story. So after we did malaria, so first of all, very recently, we've also demonstrated that you can see malaria also using without a label at all. So on the right, for example, you can see these pictures. And this is also Paul Lebel and a couple folks in BioHub also demonstrated this. With 4 or 5 nanometers, you can actually see parasites with 0 labels. So now there is no dipping, no dye itself as well. Although the problem, it adds to the cost associated with you can see that this is a very high numerical aperture for the illumination. So it increases the cost of the setup. And so for what we're doing currently, it's not worth it in terms of going completely label free, but it is valuable and interesting to have. The other thing about the platform was when we started developing and building, TB came up as a very important criteria. And so then we adapted the tool for TB. So now we can fairly regularly do TB with the same set of a scenario in sputum samples. And I'll talk a lot about this. This is kind of the SQUID platform, which is the research version of most of the Octopi platforms. And I think one of the key things that you'll notice in here is, again, letting chemistry do half the work. So this was a collaboration with Carolyn Bertozzi and a graduate student in Carolyn's lab, Mirel, had been developing these class of dyes. They're called the 3HC tree dyes. And then now they have an even better version of it, which only labels live TB. So this is another very important thing in diagnosis. If TB is transmitted through aerosols, it's transmitted by people coughing. And so you want to know whether somebody is positive, but you also want to know whether they're actually carrying some live pathogens in their cough. And that led to the deployment of this platform. And I think I'll zoom in and you can actually start seeing those are individual TB samples. And I think I might have a better image for this somewhere. TB. Let's see if I can find that picture. So TB is a very similar challenge to malaria in terms of being able to quantitatively tell TB samples apart. Yeah, I think there is a newer data set that I don't actually see. And then I think in the original paper, there are a couple other diseases that we also just demonstrate. So leishmaniasis is another one, trypanosoma. Many of these are blood parasites itself. Much easier target too, because any parasite in blood would be fair game for something like this. And one of the key things here is also now tissue scanning. So this is another example of that, in which there's lots of applications in which biopsies are made, and then after that you have to scan the tissue. And I think the purpose to highlight something like this is much more driven by this idea that, of course, we focused on making sure that the instruments work for one specific application. But then once you share that, a lot more number of applications kind of pop out that even we hadn't anticipated. And then this is what I think in the context of deployment, this is what it looks like. So this is Ethan from the lab in Uganda. We have and Rwanda. We have 2 of the instruments in Uganda now for almost 2 years. And one of the key things, although lots is changing, you can see the versions that are in the field right now. They are not in a box, they're just sitting. So lots of things go wrong. And that's kind of the direction that much of the deployment is moving, is that we really have to seal them up. Other sets of things that we learned from these was power outages. So when the instruments were out there, we would get a power outage and the scan would just stop in the middle. And then you have to restart. Even if you have battery backups at hospitals, they're not smooth. And this is actually true for a lot of medical equipment. And so one of the key things that we built afterwards now is essentially a backup battery bank. So that makes sure that you can pull the plug on it, it'll just continue to scan, and then it'll give you a warning when you have maybe less than an hour of battery left. And so this, the battery power is actually an important aspect of that. Now this is one of the sites both in Uganda, and these are all still research sites. These 2 look much better. You can see lots of other things, primarily because that's a research station. This is not the primary health center. But the one on the right is actually up north in Assam in India. And one of the big things that we've been struggling there is just the amount of rain and moisture. And so that's another aspect. The instruments are still working, but that's another factor to consider and think about, because most of the time, when we are in a research environment, you assume air conditioning and you assume a completely controlled environment. When we test them, we use as completely controlled environments, but that's not really the case. And Assam happens to be I don't know whether they still have the world record for the highest rainfall in the world. I think Cherrapunji used to be, so this is not too far from Cherrapunji. But again, I think for most of the places, building and designing for that becomes important. And then I think now this is another installment in Liberia. I think similar sets of challenges in terms of you can see that the hospital, that's Rick right there. And the hospital itself, this was a big Ebola clinic itself in Liberia. And actually, the person, Rick, in that photograph was, I think, the very first American infected with Ebola. And he was delivering a birth during the time of Ebola. And I mean, he survived through it, but he was airlifted, he was brought here. For the longest time, it's because of some of his symptoms did we actually discover that Ebola lives in the eye. So yeah, Rick is amazing in terms of the field side of the story. And then you can also just see a container ship. So much of the research unit is actually built in many of these container ships. Okay, I think I'm gonna switch. So currently what we are trying to do is on the octopi side of the story, we are trying to install these sets of instruments in a very broad range of research labs and field validation sites. And the purpose of that installation is to just really understand how these instruments arrive, what is the breadth of things we can do with these sets of tools, and also bring automated imaging and microscopy into the fold of diagnostics, because in the past that was taken away and there was this sort of semblance of the role of microscopy over time has gone down because you need trained people. And so now is the chance we can flip that around, because much of that can be done with machine learning. And machine learning is not just good for making pretty paintings and telling you what you should eat for breakfast. It can also save lives. Unfortunately, in terms of the amount of effort that gets put in on those types of applications is actually much smaller. But I think, yeah, this is one of the key aspects is imaging used to be burdensome in the past because it was all manual. But if we can flip that around, and again, you know, these are easier, lower hanging fruits than even the more complex applications, right? Reading radiology, reading, say, cancer slides. There are lots of nuances that you can make a mistake, but here you're just looking for parasites. It's much easier, low-hanging fruit. Yes. Marwan, we need a Sudanese collaborator. So if you can find us a malaria site that would be excited to do clinical work with us, that would actually be absolutely fantastic. We don't have a Sudanese collaborator yet.
Yes, I will prepare for that.
Okay, let's do a Zoom call. We can even chat sometime tonight. I think one of the big goals is to make sure that we have a majority of the countries covered, and especially because we are in this transition phase of productizing it, we would love to have someone in Sudan.
Is the deployment in the UK just like, is that an actual malaria site, or is that like a testing site?
Yeah, that is a malaria site because UK, I have a friend that does a lot of drug screening on malaria. And so one of the threads, again, is all I said was initially, I mean, the instrument is very, very general. It can do massive amount of imaging. It's incredibly robust. But then what you use that for is dependent on a function. So I'll talk a little bit about now the diversion of all kinds of other things you can deploy these class of instruments at. So we'll switch to the paradigm that we call SQUID. And the sort of one of the key things to think a little bit about is the broad idea that we started with was that we need to make scientific instruments to be much more modular and much more transformable. That was the key idea. And in order of appearance, once we finished Octopi, or we're finishing Octopi, we started realizing that we should generalize that even further. And that essentially became SQUID. So You gotta know the shortcuts for, where is the, yeah, just give me 1 second. How do you do, oh. Okay, now I don't see what folks see. What was the question, Samir?
Sorry, it was just like what, like basically what are they doing in the UK? Like I wasn't, is there actual like
Yeah.
patients that are being tested, or is it some
Yeah,
development?
what we are testing in the UK was essentially some of the parasites can actually be grown, and the UK site is really around doing drug screening, because in the earlier classes I had mentioned that for malaria, we only have one last mile, last in the line drug that's still incredibly effective. And so the goal is that we need to be screening much more rapidly. And I think this is, if you talk to the group that sprung up soup screens, this is one of the applications that they are looking at is, so they're primarily doing drug screening, which is finding newer compounds that are already pre-approved for human usage, but finding completely new uses of them. And this is a very common thing that's done, is repurposing of drugs, because it takes a long time to do the toxicity studies and other studies. But if you can find one specific molecule that has a broader application, that helps significantly in lowering the cost and just lowering overall cost of drug discovery.
That makes
And
sense.
the lab, this is Jake Baum's lab. The paper that I had shared with the soup screen that then became the name is Jake's lab had discovered an active malaria compound in a soup, in an actual ingredient of soup.
Wow.
And I'm trying to remember, do you know which culture the soup was from? Vietnamese soup. So just so you know, and just, you know, it's a really beautiful paper. It's very well done. It's actually, they requested high school students to propose components. And so Vietnamese soup came from there. They tested things in toothpaste. They tested things in all kinds of, and so again, you know, this becomes really valuable, that we are surrounded by chemicals. I mean, the chemical universe around us is massive, but clearly we, and again, it also has both good and bad implications on our health, but it's about making that, ensuring that actually something like that would work. So that was a drug screening assay.
Cool.
Okay, so I'm going to switch to squid, which is what I wanted to talk about today. Now, this is much more of our research platform that we have released. And again, it's incredibly modular. And the sets of modules, I'll kind of walk through, first of all, these sets of components. If you guys are not used to seeing these components, it's valuable to walk through this slowly. So firstly, you can see this represents an objective. Then essentially you have a set of a telescope, and this is an iconic representation of saying that you can do fluorescence, bright field, dark field by putting a filter in front of the camera. So if you were to put a filter, then you would only detect that specific light. So you can do something like fluorescence. Now, suddenly with this, one cube added to it, and the cube essentially has a glass piece that passes through, which also reflects light. So you can now see with illumination, light would come through here, bounce off, it would illuminate the sample. This is called epi-illumination. It would go through back, and then you would detect it on the sensor. Now, you can also measure something here, because a small portion of the light that you put in also passes through here, and you can detect it to optimize for you know, how much illumination are you actually providing, for example. So this is one, but now suddenly you can start saying, oh, wait a second, I can put another cube here. And this is how it becomes modular. So now the moment I've actually added another cube, you can see some of the light that's detecting is being detected can be diverted back. None of this direct light would come here, but it would bounce off. It would go on this side. It would bounce from the sample. If this is a fluorescence setup, it would come in, and a portion of that can come down. And you can choose the transmissibility of these mirrors and these 45-degree pieces put in to allow for a percentage of light to go and a percentage of light to be reflected. The other thing you can then do is also apply filters. So you can see these things are also filters. So then you can very specifically filter. And immediately you can start seeing, oh, now instead of building a 2-arm camera or a single camera, now you have 2 cameras right there. If you have 2 cameras, you can do all kinds of things. For example, do a slightly 2 focuses at the same time, 2 different planes. You can start doing volumetric things. So this is a very modular kind of a platform. And so kind of I'll jump, for example, to another case here. So now this one has, as you see, it has 3 sets of cameras, and you can see, you can start applying as many number of them. And so lots of optical capabilities can just be built in a modular way. So this goes to the optical configuration. The other sets of modules are the stage itself. So you can see for most instruments, you need automation. And so that really comes from some of these sets of stages. You can see there are 2 common stages that we build. One of them are for smaller travel. So this I would say would be around 20 to 30 millimeters, while then these longer stages are designed for much longer travel. For example, that's where you can put a well-plated. Again, just 2 common modules. Then another module that becomes important is essentially the focusing module. So this is the Z focus. This mounts on the stage. It can take a camera and an objective here. And so this module is always, you will see very commonly, is mounted on the module for the focusing of the objective. Of course, there are these cameras that I had talked about. Initially, all of these cameras were really developed for the security industry per se. Often we put telescopes in front of these cameras, and all they do is match the field of view that's coming out of the objective to the field or the size of the sensor. So all of this is doing is basically taking an image that's on the back focal plane of an objective, but it might be a really tiny image while you have a very large sensor. You just need to stretch it. So this is just an optical stretching device. It takes the focused image and then converts it into a focused image on the sensor. Now this is a laser launch. And again, these sets of components start becoming more and more elaborate. And of course, right there, you can see a souped up Raspberry Pi. There is a heat sink there. I don't know why there are 2 heat sinks. I think you can see the passive heat sink, and on top of that is the active heat sink. But then under this is the controller boards. So this is what we will use, and we'll walk through this. This has all the etch bridges, all the controls that's needed in terms of power electronics to be able to drive these sets of motors. And we'll get a chance in the lab session to build one of these, but the squids are incredibly powerful because now with this generality, we can make anything. And I really mean it. Like the number of, we last counted, we built maybe 37 or so different instruments just out of this configuration. So, I think they look like these, and every one of them is enabling totally new functionalities. And I'll walk through some of the functionalities. And of course, one of the key bits for us is it's very important that we take these instruments out of just the lab setting. So many a times, many of these pictures are not even taken here. For example, whenever you see a picture that has a round window, that's always a good indication this is on a ship. You don't make round windows. I don't know why they make round windows in ships, but so round windows, that means it's on a ship. Square windows, you are on land.
But round windows are in planes too, right?
Yeah, that's true. Yeah. It's for fracture. What does that mean? Oh, you don't want sharp corners. Yeah, that makes a lot of sense, actually. Yeah, yeah. Is that also then true for airplanes too? Yeah. That's a pretty cool idea. Like having sat on airplane windows for the longest, I never thought. So Ian answered my question for why round windows, and it's probably for fractures. So stable to much more mechanical perturbations. Okay, so I think I'm just going to give you a quick tour of the kinds of instruments that we've built with Squid. And then in the lab session, we will walk through essentially many of them. So you have a much more of a granular view of what can actually be done. And just very broadly, what we are trying to replace is this very black box kind of an approach in science. You know, the problem is whenever a technology gets commercialized, it's massively overpriced. So effectively, you know, here are just some examples of lots of platforms that incorporate, majority of it is just imaging. And they are essentially implementing one specific assay in biology. And every one of them, for them to be commercially viable at that space, starts with $500,000. While literally with these sets of modular parts for tens of thousands of dollars, we can essentially build a given capability in the open domain. And of course, much of both Squid and Octopi are open. So we help people build these sets of instruments. But this is what we are fighting against, this idea of black boxing of scientific instruments. You know, for products that are diagnostics, this makes a lot of sense. Because for the diagnostic platform, you don't want the lab technician to one day say, oh, let me just tweak this out. And then suddenly all the results are off and your certification is lost and you're not provided. When it's healthcare, it's different. But in scientific context, it becomes very valuable to have open up the capacity for scientists to then be able to say, hey, I have an idea. I'm going to try this. I'm going to try that. So I think this is kind of our answer to moving away from this to really be building open instruments. And I'm going to show you a series. This was a kind of fun for us and me and Hong Chuan to put together, where we were listing every application that we have already worked on. And the list was roughly around 40, 45 of these types of things. So I'm just going to show you some videos from each one of them. In general, again, just as a thing to remember, this is the breadth of what we have done so far in the kinds of modules. So you can see the camera options roughly from 0.3 megapixel to 200 megapixel. Frame rates for these things can even go all the way down to 10,000. These are all CMOS cameras. And then the other part, these are the total number of optical modules that we have implemented: dark field, bright field, polarization, fluorescence, lifetime. And then the thing that I was going to mention on the hardware side, I'll talk a little bit about a new module that we've been developing. It's on fluidics, which allows now you to bring chemicals so you can actually run experiments, not just a passive thing that you're taking picture of, but actually run experiments. So these are the total number of configurations. In terms of squids, this is where the current squids are. So I'm putting together a better picture of it. But one of the things that's exciting now, this is not the Octopi map. This is a squid map, which is research applications. Because again, you know, I think research capacity is a big part for us. So I've already talked quite a lot about the malaria application. This is something that you all saw. The second one we have been working on is the malaria drug resistance assay. This one is gametocyte detection. So one of the key important aspects in malaria transmission, just like TB transmission, is that there is a specific stage in which the parasite is present in someone's blood, which gets picked by a mosquito. And if that gets picked up, that's when the mosquito will be positive. Those are called gametocytes. So what's exciting is you can actually detect gametocytes in the patient's blood itself, and you can pick up people that have very high. So one challenge currently in malaria is with asymptomatic patients, there are people that just transmit malaria, but they have no symptoms of their own. So you would feel perfectly healthy, but you are a reservoir of malaria and you're also transmitting malaria. And so you have to detect in the blood whether they have gametocytes. So this is a research application which allows us to really understand what would be the propensity of the spread of a disease, for example, once you understood the number of gametocytes that are present. So here is the latest TB data set. This is a list that we just put together. So now you can see this is with a conventional dye, auramine, and what this allows us to really actually see, there are all kinds of other bacteria as well that are present, but it's fairly straightforward to actually see and detect TB from this perspective because it lights up in sputum. And then there's a fun movie I want to show you guys. I totally forgot, but I'm gonna see. You know, sometimes clinicians say funny things. So this is a lady that I met in Senegal, and she was very kind, and you know, it was a really fun conversation. Of course, this entire conversation is in French, so I don't understand except her last word. But I'll just play this for a second to give you guys a context of, you know, all of this technical work eventually has to mean something to people. And so I'll let this play. Okay. Any of you speak French? A little bit? Okay. Yeah, you guys can tell me what she's saying. So I'm just a fly in the wall watching and filming this. So just what was going on a moment before, this is in a clinic in Senegal. He has been working for the last 30 years as a microscopist. So she's the primary person that does all TB and malaria for the entire district of Frugal. So she will essentially work 10 hours a day. And she says something in the end. She said, "You are 30 years too late. I've already lost my eyes." And it was just a moment. I mean, of course, she joked about it. But what she was kind of, I think this is, sometimes you feel this tension. Oh, you're building a technology, you're taking away something from someone, not realizing at all that just because that's the standard doesn't mean it's the right standard. And I think this was a really I mean, we had lots of conversations with her much later because now she's one of the evaluators and one of the units that we're putting. But it's very interesting in these sets of scenarios to have these sets of conversations because, you know, we could build a final product and then take it out there, but we wouldn't understand who are we trying to enable. And this is very similar to the conversations that I've had with Prem and Durga, for example. The other two folks that you've also met in the class before. Again, you know, just this is what the reality of microscopy looks like in the field. And it's just not tenable when you're really trying to provide. We've got to be able to do better in terms of automation of these sets of processes. Okay, so I think this was just a little diversion, but I wanted to remind you guys of, you know, why do this to even begin with. Okay, another TB platform. You've seen this data before as well. This is again on a drug resistance side of the story. This is a new collaboration that we just started with a group in Kenya when Grace, one of the students, visited an STI clinic. So this is much more focused on sexually transmitted diseases. And one of the key things that is done in sexually transmitted disease, and again, also focused on bacterial vaginosis, is the fact that you can detect bacteria very directly. So this is all data from a squid collected, and there is a score that's essentially provided. So this is a classic case of a cell that's covered with a specific type of bacteria. And you can see in imaging is currently used as the gold standard for these sets of assays. But then again, same set of a scenario, a person sits down, makes a certain sets of diagnosis. And again, the goal is to just use the research platform to identify a broad number of applications and then flip them around to productize them in a manner that then it really becomes, because I think we have to understand the viability And this is also why squid has been very powerful for us to very rapidly. So you can see here, this one has a turret. So in the same platform, we needed to understand what magnification we would need. So this has 4 different magnifications as compared to traditionally just one. Now, this is on sickle cell disease. This is a picture of a clinic in Nepal. You can see, if I was to show you what I love about this photograph, and I don't think I have it, inside it looks like an incredible building with a lab. Outside, this building is not even painted, and it just from the outside, it looks like a building under construction. And you know what that reminds you of is they had their priorities right. They needed to get the inside done so it actually works and not care too much about what you look from the outside. And initially, I only saw the picture from the outside, and I was like, wait a second, are we sure this is the building? But it's, I mean, it's remarkable. And this is actually only possible because last year, a student that joined the Frugal Science team essentially identified a clinic in Nepal. So this is his project from the class where he's now taken octopi, and we are doing a lot of work on sickle cell disease. And there is a clinical trial that we were able to get approved in Nepal, which is ongoing, and that's Pranav. I don't know if Pranav is online today, but folks from last year would remember him. But this, I mean, it gives me a lot of joy because literally the idea started in the class, and then now he is currently in Nepal running the clinical trial. So this is ongoing. So now I'll switch a little bit to broader other types of application. This is one in serology. And I think I don't have a picture here. So let me see if I can find the multi-sero picture here. We started building something for well plates and larger fields of view for doing dengue serology. And the idea was that, you know, often enough, these types of ELISA tests, these are sandwich tests that you can do. You can detect antibodies and things in a given set of a sample, but their throughput is quite low. And so currently there is an idea that's being explored that instead of doing a single test, could you do thousands of tests simultaneously? And rather than just looking for a antibody, you could actually look for a much broader sets of things that are in a clinical sample, because you get a clinical sample just once. So it's valuable to just search for all. And again, with dengue, there are many serotypes. So there's no one dengue, and it's quite complicated that if you have dengue 1 versus dengue 4. So this is the platform that we built. We called it multi-sero. And this is some of the data from it. And you can see that in a single spot, but if these spots are really tiny, then you need microscopy. Traditionally, you don't use microscopy in this application, but if the spot keeps getting smaller and smaller, and you can also just see some of the challenges that there is bleed through between these spots because they're just spotted on a type of a substrate, and there is some amount of diffusion from the spot. So then the machines have to get better to pick up good, reliable signal from something like that itself. So there is some amount of crosstalk in this. So this is, again, this is a platform for serological assays. And then I'm going to keep going. This is what that platform essentially looks like. So now you can see this is squid. And what it's doing is, if you watch here, the well plate number is changing. And then this is the one I want you guys to use, the soup screen team, to essentially do the drug screening side, but incorporate mounting and remounting of the plates. So then you can actually run and collect this data over and over again over a long period of time. And again, all of the platform are exactly the same drivers, same sets of joysticks. You already saw this module. This was the larger set of a stage, and literally just a couple of parts, and then we can put together a kind of a system that you know, you might not know that you know how to build, but you know how to build all of these things once you kind of distill them. I already talked about lymphatic filariasis. This is a schistosomiasis system. This is much more for very high throughput imaging, but maybe I'm going to switch to, yeah, so I think, let me show you the immunophenotyping system. And This is driven by a system that we tested in Liberia recently. And the key application there is often enough you have a sample and you can have lots of different types of white blood cells in a patient. And so the problem that came to us was on Ebola survivors. It's actually very valuable to know why specific people in Ebola survived with no, I mean, there was no treatment essentially for the entire period of time. Other than IV fluids, no drugs were available to anybody. Other than people that got flown out, they were given some experimental drugs that were very effective. So it's important to know what in the immune system from a survival point of view. So NIH started and launched this big program. It's called the Ebola Survivors Program, where they give a small amount of blood, and then they are immunophenotyped in the sense that you want to know and understand what is the breadth of immune cells that they have in their blood. Up till now, all of those assays were happening at NIH. And so one of the key things that we decided to do was to build a platform that does that in Liberia. So that was the first trip. The instruments we had a couple of failures in the round one. So we're going out there again to deploy. One of the laser launches failed, and it also taught us a little bit about how to pack complex instrumentation. Because again, so what's happening here is there's a big laser engine that does 5-color illuminations. You can do very broad range of fluorescence microscopy, but there is also a plumbing system. And this plumbing system essentially brings stains to the cells, takes the picture, washes those stains off, stains it again, and continues to do that for 72 hours. So then you can have multicolor images. So this, for example, you'll see the cycle here. I'll just let this play. And this is built, you can see the 4 colors of the wavelengths, 1, 2, 3, 4. And what the system is doing is, this is sped up. It takes the 4 colors, it washes with a fluid, whatever it had stained before. It'll introduce a new stain, it'll color them, it'll take the set of pictures, wash that again, and just keep continuing that as a process. And from this, we can build essentially a map of all kinds of immune cells that might be present. But what's very valuable and important is this is also done with patients. There are no good model systems for Ebola. This is all done with humans as the direct model system. And so, this is continuing currently, and this opens up a very broad range of just immunophenotyping. So we're just finishing this paper right now, but one of the key goals is to be able to label a very broad range of cell types present in human samples. I'm going to skip this one. This is another similar example, kind of cell painting, and this is also where this is the first time we've now also incorporated a handheld robot to the microscope. So now what this can do is pick up, this is Dorna. We just got this robot. This setup is in a collaborator's lab in Sweden. This is actually a really fun robot. It's only 3K. So amongst these types of assays, it's the most cost-effective robot. It puts the plates in, it can pick up the plates, put them in incubator. So you can see incubating plates on that hood on the other side. This is the 5-channel squid system. And then, of course, combined with this allows us to do. So now there are 6 of them in Sweden right now. So this is what each one of those setups looks like. They're exactly the same. And this is very specifically for, again, both drug screening and cell painting applications. Cell painting is a general idea where, you can throw something, paint a given cell, remove that dye, add another dye. To just give you infinite number of colors, because otherwise there's a limitation in labeling that the total number of stains are only finite based on the number of channels you can detect. But if you can add and remove, then there's no limit to the number of things you can paint. This is what sort of pictures of those types of setups essentially look like. So you can see these are all of these different types of immune cells in a tissue kind of a sample. But this is all squid data. That's the fluidic system that I had mentioned, and kind of we'll see. I don't think Friday we'll get enough chance to go through this. But then again, what's very important and very useful in this platform was this piezo pump. So unlike traditional large scale pumps, now this is a solid state pump. So it works far more robustly because it doesn't have any parts that are, these are all essentially driven by piezo membranes. Another platform for bioluminescence. Now this is essentially very similar, except now we swap the camera to do a very, very high sensitivity camera. So it can detect tiniest amount of bioluminescence. Another example of that, another example of we call this the hotel for organisms. So this is a high throughput system for C. elegans. You can have tiny little hotels. And then, of course, there's a lot of work that we do in tracking side of the story. And I'm just going to show you the tracking systems. Let me see which movie this is. Yeah, so this is a volumetric imaging system. Again, very similar framework, but now you can see this field of view is jiggling. So I'll play that one more time. And what we are doing is just a quick optical trick where the optical focus itself changes, and with essentially what is called a liquid lens. And this gives you 9 different focal planes of the object simultaneously. So I'll play that one more time. When you watch this movie, it appears as if you can see 9 different focal planes simultaneously. If you could do that, then you know the 3-dimensional aspect, but we're doing that with a trick by essentially using a liquid lens. And again, all of these things would be completely different capabilities, but because we're building it modularly, you just assign and build another set of a component, and then you have the capability now goes from doing planar imaging to volumetric imaging. And then maybe the last one that I'll show you, which is a big, of course, it took a while, but is a very important one. This is probably data that Ian took. These are coral larvae being tracked. So now, while doing the volumetric imaging, we're also actually tracking the organism. So now, because of course it's an autonomous instrument, we can not only study static things, we can actually study dynamic things. And it appears that this larva is going this way, but it actually is climbing. This is a coral larva that we collect from some of our collaborators, trying to really understand how corals make decisions. So this is a type of an application in conservation. But again, the key module that makes this possible is tracking. So we are not just completely limited to a given static object. These objects can move, and then maybe I'll show another example of that in, yeah, I guess this is just the flow structure. And I think, yeah, the key bit around these sets of things are, although these are instruments that we build, they compete with anything because these are research instruments. Now we have to build them in quality that would compete with anything else that's out there. So again, just a reminder, just because it costs you less to make doesn't mean you're actually giving away. And ironically, with this type of an approach, there's far more number of things you can do with an instrument like that than you could do with something that you pay a significant amount of money for. Okay, I think maybe I'll end with this. This is high-speed tracking. And this is one of my favorite movies. So now it's pushing the limits for how fast can you track. And this is one of our favorite plankton that we have we search for. It's very difficult, like see how erratic and crazy it is. It's tintinid. And this is a good test case for us to know that the electronics can do this. So it's doing what it wants to do, but it's never going to leave the field of view. And it took many years to actually get to this point where we can do tracking that is very robust. So it's not trivial in terms of like, it just, I mean, of course, for a lot of cells and things that are not this erratic, we were able to track them for a while, but this really pushes the limits in many ways of just what's happening. So anyway, this has not been possible before, primarily. So most of the time, most of us would just sit. Yeah. Yeah, so it's a ciliate, but it's a wine glass ciliate. So what you're seeing is this ciliate is always drunk because it lives inside its own wine glass. Can you see the wine glass? It doesn't have the table, but it's that shape. And one of the key things we've been trying to understand is dynamics of this in the gravity machine for a while. They're quite abundant, we get them, but I think in the past we could not track them because they're too fast. This is one of the organisms that also does not completely is not, correlated with the gravity vector. So they do things in the wheel that are quite chaotic. And then the cilia are up there. And then when it retracts, what you see is the reversal of that cilia. And sometimes, this is not happening in this video, they will shut down their cilia and then they will sink like a rock because they are in this kind of a glassy cage. But from those types of things, you get three-dimensional trajectories because you know exactly where the object moved. Okay, I think I'm gonna end here, but kind of one of the, this is something that we have been working on for a while, but I just learned that the launch is finally happening. Squid is going to space for the first time. So this is in a parabolic flight now. We shipped this to a group in Canada a couple of years ago and we didn't hear from them. And then finally we heard, yes, the launch is on. So you can see the squid is right there. They built a whole bunch of fluidics to do certain yeast metabolism experiments. But you can see it's inside this little Tupperware type box. When it's sealed, this is how it's mounted. And you can see this is mounted to the chassis. So these are the airplanes that do the parabolic flight. You get X number of seconds. I think this is just a test case. Eventually, this will be, because in a few seconds, you can't really get biological response, turning gravity for a few seconds. But eventually, this needs to go to ISS and some other sort of really prolonged flight. Okay, yeah, I think I'll stop here. But kind of the big thing about this is we'll turn into questions and threads. The idea really is that when you start with this premise of modularity, it feels right to have scientific instruments to have the capacity of being pliable and being able to change. And I think Squid really convinced us because every single time some new application would come about, we then don't go into the catalog looking for, has somebody made that? We look whether it's already possible and what would it take. And if we all did that as a community, you can then think about like, this is just all work really pioneered by this one student in the lab. And one of the things that starts to happen is if a larger group of dialogue and scientific community changes, we would not be slaves of this scenario of, currently how there is, just like in research or just like in real life, in research also there is haves and have-nots. So lots of places have tons of tools while many other places in a scientific context have nothing. Okay, questions, comments, threads. I know we're already just out of time. Go ahead, Alankrit.
How do you come up with the biological applications for, all these modular optics, right? I mean, how do you know where the problem is? What is the process?
Yeah, I think for as a lab, one of the key things that we do is we work in parallel on lots of biological problems, and we work on parallel on lots of instrumentation problems. And so there is just a natural collision of that. We're not an instrumentation lab, or we're not a purely biology lab. We do that simultaneously. And I think, this is the whole point of this class per se in general. You can't be just you have to identify and learn how to identify problems, and you also have to learn how to solve problems. One of the skill is half half a skill, and half a skill is not sufficient. And I think, you know, we also collaborate massively. Lots of people reach out to us, but many of those applications are actually just in the lab itself.
All right, that makes sense. Thank you.
It's also a big lab. We do have a big lab. So, you know, we're like 20 people. Yeah. Other comments, questions? Yes. Mhm. Z stacks, simultaneous Z stacks, but they're off shifted in time, but they're taken so fast that, and we have them all triggered, so we know exactly when they were taken. So there is a certain time. There is another technique that we have used, which is off shifted focal planes and then simultaneously recording, but those we can just do 2 planes and they are exactly synchronous. But the volumetric it's a very simple, it came out a couple years ago. The trick is called refocusing, but not refocusing by moving something physically, which is what you normally do when you move the objective fast. But you can see objective has a mass, so that'll require accelerations that are very high. What you do is don't do it in the focal plane, you do it in the Fourier space, and you are moving essentially a very thin, these are called liquid lenses. These are membranes that are taut, but with electric fields, you can essentially oscillate them. So they go incredibly fast. And there's no, I mean, there is a moving part, but it's essentially a drum. So imagine taking a laser pointer on your drum that you play, and you play the drum and it's going to vibrate. So it's exploiting the dynamics of that membrane. Yeah, it's a really powerful component. And then what that allows us to do is both do volumetric imaging, and then the other thing that it was already originally invented by Philips to do displays, but you know, LCDs are very hard to beat, so they didn't find a very good application in displays. But originally the idea was passive displays that don't use power, right? For our displays, every pixel to remain on, we use power. Reflective displays like e-ink and other things don't use that. And so this was an idea that you could have membrane in different states, but it remains in that one state. But I think, yeah, this is a better application. Subhir.
Yeah, quick question, and maybe a silly question. You mentioned the use of the CD drive as the motors and the way. So CD is also spiral scanning, right?
Yeah.
And is there any merit to thinking,
Yeah,
trying to use that as a microscopy setup?
absolutely. No, I think I'm glad you said this. And actually, the very original, I will find some pictures from Madagascar. The very original, when Hong Chuan was a rotation student, in the rotation, he primarily, essentially, we bought around 200 different CD drives. We reverse engineer all of them. And then the thread that we were really looking at is, instead of linear drives, rotary as a mechanism for scanning. I think eventually we went away to XY scanning much more from many of the platforms in which we do the assays are also focused on glass slides and these well plates.
I see.
We wanted the imaging assays to be compatible to the most traditional, rather than the device be dependent on some consumable. And so I think for malaria, it made sense to do a lot of things with essentially with a single XY scan. But there are a couple other applications that we've been looking at where we do rotation, and especially rotation, then we can do much faster, because
and
there's no stopping every few minutes. So from an acceleration point of view, it's just a steady state scanning.
Right. And also, if you don't want to build an image as an outcome, right? It could be.
Absolutely. Yeah, most of these applications, image as an outcome goes away because you just want to get to the answer. And so from a
Yeah.
detection point of view, it's very useful. Yeah, I think I have this, for the longest while, we've tested this a couple of times for the both spiral and cylindrical sampling. And we have
Okay.
one optical system, which I didn't talk about. We actually have that rotation incorporated. This is when Benedict was visiting, we finally
Mhm.
finished that. So there is one rotational scanning that we have. But I think overall, for the lab session for this week, what I would like everybody, while in the lab here, we'll be building squids. I would like everybody who's online to go either buy or find optical DVD or CD drives, and you guys will be disassembling those to realize just how, and actually the entire squid, all of that started with us just trying to understand CD drives and how much optics they fit in this tiny configuration. We moved away from it because it's not so modular and it didn't give us the kind of degrees of freedom. But for a lab exercise, what I would like all of you to do is open and try to understand and explain to each other how these DVD drives work. And then, we did hack through, so we have all the logic board codes for how to talk to DVD drives. So eventually, if somebody picked up a project on those, that would actually be fun. Okay, it's 10:05. I ran a little bit late, so we'll continue this discussion on Thursday. But just as a reminder, March 14 and 16th are the class presentations. 10-minute talks and infinite amount of questions. We'll figure out how we'll make that work. It's in the past, what we've also done is have people record a video of their talk and then have mostly time for discussions. I'm thinking just let's do live presentations. I don't know how people feel about live presentations. Okay, let's do live presentations. Yeah, I think I was leaning, I don't think it saves any time because if people wouldn't have watched those videos, yeah, let's just do live presentations. But I think, yeah, if a team, and Alankrit, let's do the Fred Park first thing, 8:30 Thursday.
Okay, all right.
Yeah. I think it's just, I want to make sure that people will have to run to classes and everything.
All right.
Yeah.
Thank-
Okay. So we'll see you Thursday, and then a big lab session on this Friday. And I think after that, all lab sessions will just be project work. I think, yeah, there is a little downside. I mean, on one hand, it's valuable for you all to see broad things, but it's also, on the other hand, you need to focus on your projects. Okay. Bye, everyone.